All right, ladies and gentlemen, welcome to our lesson on protein synthesis. So we're going to talk about how proteins are made. So just a few terms to clarify. When I talk about the genome, I'm talking about all of the genes or all of the collective DNA that an organism has. So what you are looking at is an image of the human genome. These are the 23 pairs of chromosomes that make up our genome. Before we move further, I'm going to introduce you to what is called the central dogma of genetics. A dogma is a super important kind of tenet or a basic kind of fundamental founding principle of something, and this is one of the most important, one of the most central ideas in genetics, that information always flows from DNA to RNA to protein. We can add in a few things to that. We can add in details like which particular RNA is carrying the information. So for example, information goes from DNA to mRNA, and then the mRNA transfers information to tRNA, which then creates a polypeptide or a chain of amino acids, which can then be folded into a mature protein, and then all of that leads to the expression of a trait, which will become important when we talk about genetics, because when we're talking about genetics, we're going to look at the end result of this whole process of making proteins. This is another way to represent that, just showing you have your information going from a DNA to RNA. In this case, they're specifically showing you mRNA to the amino acid sequence of a protein. So if you see something like AAs in some sort of genetics problem or a DNA question, that is talking about an amino acid, which are the building blocks of proteins. We'll talk about that in more detail when we talk about biochemistry. Now, this process in prokaryotes is different than how it happens in eukaryotes. As always, that any processes that happen in eukaryotes are generally more complex than those happening in prokaryotes. The major difference here is that in prokaryotes, you do not have a nucleus, so you can actually couple the two processes that make up protein synthesis. You can make an mRNA, which is shown here, and while that mRNA is being created, you can actually use the instructions in it to make a protein. So here you have ribosomes that are creating a protein based on the sequence of an mRNA, whereas in a eukaryote, those two processes are separated by this boundary of the nucleus. This allows for a few things, a few extra steps to take place, which we'll talk about later. You can actually process your mRNA and make changes to it before it goes out into the cytoplasm and gets translated. So, like I said, it's much more complicated of a process in eukaryotes. Like I also said, we'll talk about that in more detail later. You have a few things that can happen. So, for example, once you make an mRNA, you can cut out these sequences called introns. You can actually make alternate versions of a protein from a single mRNA, and then you also add some things on. So, for example, you add a 5' cap and a poly A tail at the 3' end, and I'll talk about the functions of those later. So let's talk transcription first. Transcription is performed by a single enzyme called RNA polymerase. RNA polymerase does a couple of jobs. One thing it does is it will unwind the DNA double helix so that you have access to the nucleotide sequence of the template strand, and it will then add on complementary RNA nucleotides. And at the very end, you're going to spit out an mRNA copy of your original DNA template. So you have transcription happening in this direction. As the DNA passes through the RNA polymerase, after it leaves or exits the RNA polymerase, it rewinds up. So you're not separating the DNA for long, you're just separating it just long enough for you to get in and copy the sequence of nucleotides or make an mRNA version. So it's different from DNA replication. Remember, in DNA replication, you start unwinding the DNA double helix, and when you unwind it, it's not going to come back together. You're going to have two strands of DNA, or two new molecules of DNA that are being made. So that's a kind of different process. Whereas during transcription, you are going to wind that DNA back up. You're going to read it, then you're going to rewind it. Like I said, we're going to get to the complexity now of RNA pre-processing or pre-mRNA processing that happens in eukaryotic cells. So this creates a mature mRNA that can then be translated by a ribosome outside of the nucleus. Without these steps, you are not going to have a translatable mRNA. So what we're going to learn as we get closer to when we talk about actual translation with a ribosome is that every single protein, every single mRNA is going to start out with a single sequence, AUG, which then is going to code for a methionine to start out every protein. But you have an additional signal that's added to the beginning of an mRNA to help kind of initiate that complex with the ribosome to start translating. So it kind of helps everything get assembled together to start translating this sequence. And so what you're adding is a 5' cap. It's also called a methyl G cap because it is a guanine that's been modified with an addition of this CH3 right here, which is a methyl group. Another thing that you do is there are coding sequences and non-coding sequences. The non-coding sequences that are called introns are removed. We call that process intron excision. You can also call it splicing. And then finally, you add a poly A tail. The significance of this poly A tail is it helps kind of serve as a timer. Notice that it says you have anywhere between 150 and 250 adenines added to the end of an mRNA. Well, you have these enzymes that are floating around in the cytoplasm, and they'll start eating away at the 3' end of an mRNA. And if you want that mRNA to last longer before it gets completely degraded, you're going to put a longer poly A tail on it. So let's say that you had this mRNA. So you have your methyl G, and then you have, let's say this box segment is your poly A tail. Then you have the exact same mRNA, but instead you have a much shorter poly A tail. This guy right here, the bottom one, is going to get degraded faster because you'll start having enzymes eat away at that poly A tail. When the poly A tail is completely gone, it's going to be able to start eating up this entire transcript, which is going to be gone, at which point your cell is no longer making that protein. However, this top sequence right here, it has a longer poly A tail, which means it takes longer for these nucleases, the enzymes that break down nucleic acids, to break down this mRNA, and so that mRNA lasts longer. This gives you a little extra control over how long you're going to be transcribing a gene. So if you need a protein to be made for a very short period of time, you would put a shorter poly A tail on it. On the other hand, if you need that protein to be made for a long period of time, you need it to be made for hours or days, you would put a longer poly A tail on it. That way it will stick around longer and it can be translated for a longer period of time. So let's talk introns and exons real quick. Introns are these non-coding regions of a DNA and then RNA that are not included in a final mature mRNA. The coolest thing about introns is that you can actually do what are called alternate splices. So for example, I could make a protein where I take out not only this intron, but I take out everything from that intron to this intron, which would remove this green sequence, which would make a different mRNA, and which would make a different protein. And depending on which introns I splice out and which exons get taken out with those, I can make different proteins with slightly different functions from the same gene. This kind of helps answer a large question that we used to have about genetics, and that is when we looked at the sheer amount of proteins that were being created in the human body, when we looked at the sheer size of the genome, early estimates for the number of genes that we have in our genome were upwards of 80 to 100,000. But as we've started sequencing more and more of the genome and identifying more and more of the genes that are found in there, we found the number is actually much closer to 20,000. So you only have about 20,000 genes functioning to keep you alive. Well, how does that account for the large variety of proteins that we make? You have something like these alternate splices. The splicing is done by a complex that includes protein and some RNA, and it is called a spliceosome. So it's going to take out the introns, and then it's also going to ligate or rejoin those exons to each other to form a mature mRNA. So here are some alternate splicings. So you have a gene with 5 different exons. Depending on how you splice it, like which introns you remove and whether you remove some exons with them, you can make different proteins. So let's say you have this initial splicing, so we're going to call it splice 1. It has all of the exons contained in it, so it has all of these different regions, and these regions have different functions. So these coiling regions right here could be indicative of part of a transmembrane sequence, so an area that goes across the cell membrane. Then if you take out some of the exons, so for example, you take out exon 3, which probably is this portion right here, you can make a different protein. So now let's say this is also sitting across the cell membrane. You've now removed a large chunk that would interact with stuff inside of the cell. On the other hand, let's say you take out exon 4 instead, you still have this large intracellular domain that's interacting with molecules on the inside of the cell, but you have lost this kind of loop that was going across the membrane. And this area right here, which is coded for by exon 4, could be a binding site for a receptor, could be a place where you have interactions with molecules outside of the cell. So by removing a single exon, you can change the function of the protein without having to have a completely separate gene. All right, so already talked about the functions of RNA capping, and you're well able to read that. The addition of a poly A tail is also called polyadenylation because you're adding a bunch of adenines to the end, and those adenines, like in other molecular forms, are also called adenylyls. Like there's another molecule we'll talk about later called adenylyl cyclase, which has a very similar structure. And we've also talked about the function of that. But there are multiple types of RNAs, as we've talked about before. Ribosomal RNA is involved in the structure and function of a ribosome. You can see there's a mixture of both RNA and protein that makes up a ribosome. tRNA, on the other hand, is pure RNA, no protein. It has a very specific 3-dimensional shape. You can see it represented in multiple ways. The way that's kind of, I guess, most similar to the model that I've created for us to use in class is this 2-dimensional one where you have an anticodon sticking out at the end, and you kind of see this kind of these double-stranded regions. In reality, the 3-dimensional shape almost looks kind of like, I don't know, an upside-down L. And you have an anticodon still sticking out the bottom. Then you can have an even more stylized version, this really simplified version that just kind of lets you see where an amino acid attaches and where the anticodon is. So let's talk about translation. Keep in mind that nucleic acids are read a single codon at a time. Each codon has 3 nucleotides in it. And each one of those codons is going to stand for a particular amino acid that's going to be put into a polypeptide or a protein. So each codon stands for a particular amino acid. So in this case, we have 7 codons in an mRNA. We start out with our first codon, which always tends to be AUG, and that codes for the amino acid methionine. And then each of the other codons codes for a specific amino acid, with one exception. That is, this final codon is a stop codon, and that is saying that is going to be a message to the ribosome: disassemble, stop making a protein. So wherever you have a stop codon, that's where you stop making the protein, and it doesn't code for an amino acid. So there are multiple steps to translation. What's going to happen is first, you're going to have an mRNA. It's going to have a start codon of AUG. You're going to have the appropriate tRNA attached, which is going to have a methionine attached to the other end. And when that binds to an mRNA, the ribosome is going to attach around that. So you're going to have the large and the small subunits of the ribosome attach around that. Notice you have multiple binding sites inside of a ribosome. And each of those are where a tRNA can bind. We're not going to go into details about what each of those sites is responsible for or the name of it. I just wanted to kind of point that out because, as you see in the next step of elongation, you are going to have different tRNAs come into those sites. So we're going to start up here. And we're going to look at, this is our first picture. So we have a tRNA that's already stuck in this ribosome, and it has an amino acid chain attached to it. Another tRNA with another amino acid, which has a matching anticodon, is going to come into this next open binding site. And once it kind of locks in, that's going to cause the ribosome to transfer this polypeptide chain from the previous tRNA to the next one. And we did model this in class. So then it's going to be bound here. And notice now we have a longer polypeptide chain. The previous tRNA that was here is going to be freed of its amino acid, and the entire ribosome is going to shift over along the mRNA one. When that happens, the mRNA is almost feeding through, going towards the left of the screen. That tRNA that no longer has an amino acid attached to it moves to the next spot and is effectively kicked out. Then our tRNA that carries that polypeptide chain is now going to be in this middle site, ready for another tRNA to come in so that you can keep elongating the chain. This happens until, there are all the steps. This is going to happen until you get a stop codon come in. When a stop codon comes in, you're going to have a separate tRNA that's a release factor. It's got an anticodon that matches with that stop codon, and when it binds, that signals everything to break apart. The large and small subunits of the ribosome are going to separate, that mRNA is going to separate, and the polypeptide chain is going to be released. So at that point, the mRNA can be read by other ribosomes and be used to make more protein. That ribosome can either reassemble on that same mRNA and start making that same protein again, or it can assemble around another mRNA and make another protein. It just kind of depends on whether that mRNA is degraded or not and how far that ribosome needs to move. So at this point, you have a polypeptide, and when we get into biochemistry, we'll talk in more detail about what happens to that polypeptide before it becomes a mature protein. So one of the coolest discoveries that scientists, biologists have made is they have figured out the actual genetic code. So what each codon of RNA stands for. So we have 3 letters in a codon. We have 4 possible letters, an A, U, a G, or a C. And so when you calculate all the possible combinations, you can make 64 possible codons. There are, however, only about 20 amino acids that are naturally found in most living creatures. Because of this, you have what's called redundancy, which is that there are some codons that you might have more than one codon representing the same amino acid. This code is universal amongst almost all living creatures. There are only a few bacteria that have 1 or 2 variations in this genetic code. So this is a sign that this genetic code evolved very early in life, and the only living creatures whose offspring survived until today all used pretty much the same genetic code. So if you've forgotten how to read this translation chart, note that we have, let's say we had a codon of UGC that we were trying to translate, and we want to see what amino acid is represented by that code. Well, we look first at the area which says first base. That first base for us is a U, which means we're looking within this block of the codon chart. Notice every single codon in that part of the chart starts with the nucleotide U. Then our second base is G, so we'd look for our G. It's that final column there, and so everything within that column, you'll notice, has G as the second base. What we're going to look at is the intersection of those two. When we look at the intersection of those two, we are looking specifically at this box right here, and you'll notice all of those codons start with UG. Then we need to look at our third base. Our third base is C, so within that one box where everything starts with UG, we're going to look and see where is there a C. Turns out the second one down is a C, which means that our amino acid is represented, the amino acid that UGC codes for is that amino acid there, cysteine. Just a note, all of these terms, like the names of the different nitrogen bases or the names of these amino acids, all tend to rhyme with the word "sine" or "tine." So for example, you pronounce them phenylalanine, serine, tyrosine, cysteine, leucine, proline, glutamine, etc. So I was pointing out some redundancy. You'll notice I've kind of color-coded everything, so any codons that code for the same amino acid should be the same color. So you'll notice some of these amino acids have a lot of codons that code for them. For example, leucine has 6 different codons, whereas there are other amino acids like methionine or tryptophan that only have 1 codon that codes for them. Notice we also have 3 different stop codons. This redundancy is really key because it lets you see that you could make a mutation in DNA that changes one nitrogen base but does not change the identity of the amino acid. So for example, let's say that the original codon on an RNA sequence is AUU. AUU codes for isoleucine. If that mutated to AUA, that would still code for the same amino acid. So you could have a mutation and not have it affect the protein that's being created. Or if, for example, you had a mutation from UUA, and UUA originally codes for leucine, if it mutated to CUA, notice we did not change the amino acid. So that redundancy kind of allows for a few mistakes when you're making copies of DNA without necessarily messing up a protein and killing an organism. There are some exceptions to this central dogma. The biggest one are usually viruses, which will contain a single-stranded RNA as their genetic code. They will also usually contain an enzyme called reverse transcriptase, which makes a double-stranded DNA copy. So this process is reverse transcription because instead of going from a DNA to RNA, you are going in the opposite direction and you are going from RNA to DNA. However, your body does try to have defenses against reverse transcriptase or anything that's making DNA copies of RNA, because the only things that really do that are viruses, and if you are a cell, you do not want to get infected by a virus. All right, there goes our second lesson, and we'll have our third one up soon.